Hello, good morning, class. We are going to finish off lecture 8 in the first 15-20 minutes, and then we're going to go ahead and get started on lecture 9, right? So last time we did all the different types of inheritance pattern questions, monohybrid, dihybrid, incomplete dominance, codominance, as well as sex-linked disease. So I just want to ask if anybody want me to take up any particular questions from the study guide. Now would be a good time to let me know. I'll pull the study guide up for you to see. There it is. Is there any? I mean, I posed the answer, but are there any particular questions you want me to take up? I'll give you a moment to look through it and let me know. All right. Is there anything at all? Okay, well, if you found anything, please let me know, and we can take it up. Okay, just leave it in the chat box. Now, I do want to take up this question with you. This is something we haven't learned yet, so we'll talk about it, and that would be the last thing that we need to learn for lecture 8. So these are called pedigrees, which basically shows the relationship between different family members. Squares are male, circles are female, and the horizontal line means they are going to have kids together, and these are going to be their children, basically. Okay, so that's one generation, that's the second generation. The generations are numbered using Roman numerals. So first generation, second generation, and each individual in a particular generation is numbered using, regular numbers. So this person here is called 1, 2. Right? Like this person there is called 2, 3. Right? So that's how you refer to them. If you need to, or in a question, they would be like, find the genotype of individual 2, 3. Then you know they are talking about this guy. Okay, so you have to be familiar with the description. Now, what we are asking you to do here in this question is to determine whether the inheritance pattern is autosomal dominant or autosomal recessive. Okay, autosomal dominant or recessive. So let's take a look at this first one here. Okay. You can just guess. Okay. You can guess and see if it works out, right? And if it doesn't, then it's the other one. So let's say, like my guess, okay? We're gonna guess it is dominant. Okay, dominant. So like big A means the person's gonna have the condition, right? So this is the only person in the whole family that has the trait. Okay? And if our guess is that the inheritance pattern is dominant, then she must have a big A. I don't know what the other one is. It could be big A, or big A, small a. But if we assume this is dominant, then she must have a big A. Now, where did the big A come from? Well, it could be from the dad or from the mom, which means one of them must also have a big A. But that's not possible because if the dad or the mom has the big A, they must also have the condition. But they don't have the condition. They're not shaded over here, right? They're not shaded. So that tells me this cannot be a dominant pattern. Okay, so guess is dominant, okay, but the daughter, right, 2, 2 has to inherit big A from 1, 1 or 1, 2, but neither of them has the condition. Therefore, not dominant, right? So it must be recessive. Must be recessive. So how does it work for recessive? Okay, let's find out. If it's recessive, then 2, 2 must be little a, little a. Right? And that means the parents who don't have the condition, they must be heterozygous. So if you do the Punnett square, you'll find out there's a 25% chance that they will be able to produce a child with the conditions that they don't have. So here's the key thing to remember. Key thing to remember. Okay, you're looking for this kind of pattern. Okay? You're looking for a situation where if the parents don't have it, but one of the children does, then it has to be a recessive trait. Yeah, I'll write down for you. Okay. Parents, and then like whether it's a son or daughter doesn't matter. Right? The key is if both parents don't have the condition, but one of their children has it, then it is recessive. Okay. Then the condition is recessive. Okay. I just showed you earlier, right, why this wouldn't work for dominant. Right? So if you see this, it's recessive. Question? Okay. Let's take a look at the second one here. We can take a guess again. Okay, guess. What's our guess this time? Let's guess it is recessive this time. So if it's recessive, what must be the genotype of those two people? Anybody? if this was a recessive inheritance pattern, what must be the genotype of this person? He has it. He has the condition. So if it's recessive, what would his genotype be? That's okay. So he would be little a, little a. Okay? And then the spouse, the wife, also little a, if this was recessive. But you will see this is not possible. If they are both little a, then all of their kids should also be little a. But they're not. They don't have the condition. Okay? So our guess is wrong. But none of the second generation, none of the children, I guess you can say, from of 1, 1 and 1, 2 has the condition. Therefore, not recessive. Okay. So is it dominant? Well, let's check it out. If it's dominant, then this must be one big A, one big A. Now they wouldn't be big A, big A, big A, big A, because again, all of them would have the condition, right? So it's going to be heterozygous, and all these people would be small a, small a. All right. So you're looking for this kind of pattern. Right? If the parents have it and the kids don't, um, then you know. Then you know is going to be recessive. Sorry, dominant. So it's the opposite. Okay. So that leaves us with one last one. This one over here. So based on what I just told you, can you tell me whether it's dominant or recessive? You should be able to tell right away. Just based on the patterns I was telling you about. Anybody? Is this dominant or recessive? Dominant. Well, anyone else? So let's see. We have parents who don't have it, but the kid does. And then here is another situation where the parents don't have it and the kid uh, has it, right? So that's like same as this pattern over here. Right? So like whenever the parents don't have it and the kids have it, it has to be recessive. Right? Let me explain why it cannot be dominant. If this was dominant, then this individual would have a big A, right? And where does the big A come from? Has to be from one of the parents. But if that's the case, if let's say the big A came from the dad would have been shaded because it's a dominant trait, right? but he's not shaded. Right? That's why it cannot be dominant. This has to be recessive. Okay, has to be recessive. So the parents are heterozygous, and then like this. Right? I mean, we can figure out the other ones as well. This one, they don't have it. So like it's capital A, but we don't know what the other one is. Same thing over here. These all have it. And then these kids, they don't have the condition, so they have to be heterozygous because the father can only pass on small a, right? So that's what it is. Okay, let me show you the poll question over here. You know what, let me just copy it to the other page so we can do it together. Okay, the pedigree below shows family members who exhibit a dominant trait. Dominant trait. What is the probability that the children of 4-3 and 4-4 will have a dominant trait? So this is 4-3 and 4-4. Generation 4, individual 3 and 4. That's what we're trying to figure out. So because it's a dominant trait, we can say like big W, right, is dominant, and then small w is recessive. Okay. So number 3 here, that is someone who has the condition, so he must be big W something. Number 4 does not have the condition, so she must be little w, little w. Let's take a closer look at number 3 here. We trace back to his parents. His mother does not have the condition, so she must be little w, little w. Okay? It doesn't really matter what his dad is, but because the mother is homozygous, she would pass on one of the small w to this guy. All right. So now we're going to go ahead and do the Punnett square. That's individual 4-3. That's individual 4-4. And if you put them together, you will see that 50% of them is going to have the trait. Okay? That's all. Any questions at all? That's it for lecture 8. Here, let me just try to fix my screen here. Something is not quite right. Okay. Can I explain the last question again? Like this one that we just did with the W? Okay. So let's try again. The question tells us that the trait is dominant, which means if the person shows the condition, he or she is gonna have a capital W in their gene, okay, in their genome. So let's take a look at this person right here. Okay, this person right there. He has the condition, right? So he must have at least one big W. But we don't know if he's homozygous, which is big W, or heterozygous, which is big W, small w. The wife is she doesn't have the trait, right? She's not shaded in, right? So that's how we know 100% this person is little w, little w. So far, okay? Now we have to figure out what the other one is. Like, is it a big W? Is this a small w? We don't know. That's why we have to trace back to his parents. Okay? So this is his father and this is his mother. The mother is not shaded. She does not have the trait. So it must be little w, little w. And since the mother only has little w in her gene, she can only pass that on to her children, right? So you will take one of these little w's and then you will pass it on to the next generation. So that's how I know this person, number 43, he is heterozygous. That's how we know. And then you create the Punnett square. All right. Okay. So the quiz is going to be next week. I think we can keep it on Tuesday. But it's a difficult quiz because like there are a lot of these problems, and there's also lecture 9 that we even started learning yet. So I am gonna give you two tries for this quiz. You do it first time, and then you can see your mark. And if you don't like it, you can do it again. Right? It's the best mark that counts. Right? So like let's say you get 80% on the first try. You think you can get a little bit higher, but then after you do the second time, you only got 75. That's okay. You can keep your 80. Okay, but if you get 90 on the second time, then you get to keep the 90. All right. So everybody get 2 tries, and keep your best score. Hopefully that will help you a little bit for quiz number 4. All right. So let's take a look at lecture 9. Hold on, my screen, like I said, is a little bit strange. I need to see if this works. Okay, there it is. All right, number 9, as I keep on telling you, is the hardest lesson of the course because there's a lot of concepts, a lot of moving pieces, and then there are some problem solving as well. So we got to make sure that we are in this together, that if you don't understand, you have to stop me and ask. What is biotechnology? It's quite simple. Biotechnology is, as the name implies, technology that's based on biology. So after learning about all the things that was in the course so far, right, like the different organelles, the process of transcription, translation, understanding DNA replication, we try to control some of these processes. We try to change them and harness their mechanisms so that we can develop technologies and products which will ultimately benefit human lives. That's basically the goal of biotechnology. Okay, so we alter some of these processes that we understand, like cellular, like DNA replication. We try to understand that, and we try to change it, and then we try to put it to use so that we create some kind of technology that would help improve lives. So the first thing we are going to learn, it probably is the last thing we're going to be learning as well this for today, because this is like so much, is recombinant DNA. Recombinant DNA. Recombinant DNA refers to the situation where you take DNA from different organisms and then you put them together in the same sequence. Okay, so the definition written here is a fragment of DNA composed of sequences originating from at least two different sources. Sources as in like organisms. It could be from like humans into bacteria. It could be like from flowers into animals, what have you. The result is recombinant DNA. So that has application in medicine and agriculture, to name a few examples. There are many other applications as well. So this here is a jellyfish. Jellyfish, if you've ever been to the aquarium or even the zoo, they have usually a jellyfish tank, and it's pretty. They glow and give off this nice fluorescence. And the protein that allows them to do it is called GFP, green fluorescent protein. So what scientists have done is they took the GFP and then they put it into mice, okay? And then the mice, they glow in the dark. And you see these green mice? That's because they have the GFP protein. So that's an example of recombinant DNA. You might wonder, why would we want to have a mouse that glows in the dark? Actually, there are a lot of applications for that, but you can look it up yourself. Okay, we don't have time to go into details. They didn't just do it for fun. Okay, like there are actual research benefit for the mouse to be glowing like that. So I'm gonna go through two examples with you, and believe it or not, it's gonna take the whole class of how recombinant DNA can be used in agriculture as well as medicine. Let's start with agriculture. This one is short. We'll be done in like 5 minutes probably for agriculture. So there is something called golden rice. Golden rice, right? Golden rice was invented to help solve a condition known as vitamin A deficiency. Okay, and this is very, very common in countries who does not have access to food that are rich in vitamin A. Okay, and one of the problem that arises as a result of vitamin A deficiency is that it could cause nighttime blindness. Okay, so in order for us to see, we actually need to make this molecule in the eye, in the cells of the eyes, and we kind of need vitamin A as a starting material to make that. So when there isn't enough vitamin A, when it's dark, you can't see, you have nighttime blindness. And if this continues, it could lead to permanent blindness in kids. Right? So vitamin A deficiency is the leading cause of preventable. It's preventable because like they just need vitamin A, right? It's not something that they're born with. It's not like there's an injury or something like that. It's preventable childhood blindness. Deficiency in vitamin A also leads to lower immunities combined with other things like low vaccination rate that could actually spark an endemic for infectious diseases. In some countries, each year, it is estimated that vitamin A deficiency is responsible for 670,000 deaths in children under the age of 5 and cause an additional 500,000 cases of irreversible childhood blindness. So it's a serious situation. This is a heat map that shows you how prevalent this problem is in different parts of the world. And you can see, in North America, for example, like where we live, in countries that, the so-called developed countries, right? This is not as much of a problem because, access to food that are rich in vitamin A is just available everywhere. Like people take it for granted. But in some of the countries who are still developing, if you would, that would be quite a bit of a problem. So that's why people invented golden rice. Okay? Golden rice is like regular rice, but it is engineered to create something called beta carotene. which is a building block for vitamin A. So if you, because rice is a staple food in a lot of these countries, right, where they're having the deficiency in vitamin A. So if you add that to the rice, when they eat the rice, they would be getting the vitamin A as well. Not directly, they would be getting the beta carotene, which then allows their body to make vitamin A, right? Have your mother or like, have you ever tell your kids, or have your mother and dad ever tell you that certain food is good for your eyes? What am I talking about? Anybody heard of that before? Like, eat your blank because it's good for your eyes. What is that thing?
Carrot.
Carrot? Yeah, that's
Yeah.
right. Hmm. So what makes carrot orange is actually the beta carotene, right? And that's why the golden rice you can see in this picture is kind of, orangey yellow, right? Right? So that's what it is. But rice on their own do not naturally produce beta carotene, right? That's why it's a recombinant product. To do that, they have to take one gene from daffodils. Daffodils is like this kind of flower right here. And the gene they took is called Psy, Psy. You don't have to memorize that. And then there are some soil bacteria that they extract another gene called CRTL, and they put this into the rice. Right? And that allows the rice to create the beta carotene. Right? So that's a recombinant product because it not only does it have like the rice DNA, but it also have genetic material from the flower as well as from the bacteria. So there are like many GMOs, right? We call these genetically modified organism, GMOs. There are some controversies surrounding them. Some are valid, in my opinion. Some are valid points. Others are just people who don't understand the science, and the argument is not legitimate. Okay, but here are some controversies that I personally think are valid points. So first of all, it was not a smooth launch at the early stage when it was originally created. It turns out the rice does not have sufficient amount of vitamin A, so that's a problem, right? Like the whole point was to get enough of the beta carotene so that you can make enough vitamin A. But it turns out it didn't have enough. It didn't make an impact. So the problem was solved by developing a new strain. So they tweak it a little bit, and there is something called golden rice 2, which contains 33 times more beta carotene, which translates to more vitamin A, right? There are opposition from groups like Greenpeace, where they opposes the use of any patent genetically modified organisms. Patent means the company who developed the rice, they owns the technology, right? Nobody else can like replicate it. They are the only source of it, right? Legally speaking. And the cultivation of golden rice it would be one of these situations. They claim that it would open the doors to more widespread use of GMOs. Okay. Some worry about corporate control of agriculture in poor countries, typically where golden rice is grown, right? So like they want to have these autonomies. They don't want like big corporates controlling what kind of food is distributed in these countries, right? Too much power from corporation might have outcomes that are unintended, right? They might have like, for example, like political influences, right? And that's something that people don't necessarily want. And some argue it is sufficient to get vitamin A from other food sources. Why eat the GMOs, right? Just ship them the food that they need to get the vitamin A. But it's important to understand, um, rice is a stable product that can be stored for extended period of time. Whereas, like, take carrots, for example, like you can ship a whole ship of these carrots to this country and expect it to stay good, right? Okay, so and it's expensive to do so as well. Anyway, so these are some controversies, and that's an example of how recombinant DNA can be used to create GMOs that will help solve some serious problems. In this case, blindness associated to vitamin A deficiency. And in Canada, we actually have crops that are GMOs. tomatoes. Like, if you like tomatoes, if you enjoy having some tomatoes in your burgers or like add it to your salad, tomatoes are available all season long. Sure, some of them are grown in greenhouses, but the ones that are grown outside, they have to make them more adaptable to cold weather. So they incorporate some cold-resistant proteins that are from tuna fish, put it into the tomato, and it makes them frostbite resistant. All right, so that's it for agriculture. Now we're gonna switch and talk about how recombinant DNA can be used in medicine. And the example I'm going to show you is insulin. Okay. So insulin is made by your pancreas. Okay, made in pancreas. If you don't know where your pancreas is, it is on the let me see, on your left side. All right. Okay. This is your diaphragm, guy, and this is where your stomach is. This is where your liver is. The stomach, and then behind, a little bit behind the stomach, that's where you're gonna find your pancreas. If you don't know, that's okay. We're gonna learn that in next semester, anyways. So there are two main forms of diabetes, type 1 and type 2. The one that most people are familiar with is the type 2 diabetes, where it's related to things like obesity, lifestyle, and in those people who suffer from type 2 diabetes, their insulin level is actually fine. In fact, it's like higher than normal. So if you give them insulin, it's not going to fix the problem. Okay, it's sometimes going to make it worse. So for people with type 1 diabetes, that's a situation where they are born with perhaps an autoimmune disease where their immune system attacks their pancreas, which cause them to not be able to make insulin. Okay, so we need insulin to help absorb the sugar from our blood. So after you eat a meal, all those glucose that are in your bloodstream needs to go into your cell so you can do cellular respiration. But it doesn't happen on its own. You need to have this insulin. All right. So without insulin, people can actually, it's a life-threatening situation. So here are two scientists, Frederick Banting and Charles Best. They are Canadians who were credited for the discovery of insulin, the isolation of insulin. Nowadays, insulins are primarily commercially available, and they are actually made using biotechnology. But before that was possible, the insulins were actually extracted from the pancreas of like calves and pigs, and then they would give it to people who need it. So what are some potential problems with using insulins from cows and pigs on humans? What do you think? What are some disadvantages and drawbacks and concerns related to this?
Blood sugar.
Sorry, what was that again?
What did you say? What was your question?
Question. Okay, my question is that before we can make insulin in a lab, the only way to get insulin and give it to people who have diabetes is to take the insulin that are produced in cows and pigs, and they purify it and give it to people. So my question to you guys is, what's the problem with that? What are some potential problems? Well, after all, they are from cows and pigs, and it might not be. It is not. I can tell you, it is not exactly the same as the insulin that is produced in the human bodies, right? So the efficacy, right, like how effective it is lowered, right? It's lower efficacy, right? And it takes a lot of work to isolate these insulins from the animals, and make sure that it's purified and get enough of it to be therapeutic. That's another concern. So it's like not very cost effective. And for religious reason, people might not want to have insulin that was extracted from like pigs, for example, and be injected into them, right? So there are many drawbacks of those situations. And until the late 20th century, this was the way to go. Yeah, but then, thanks to biotechnology, we were able to make it in a lab now using recombinant technology, right, that I'm going to show you. So in order to do this, in order to make insulin, we have to do 4 steps. Just going to read it to you first, and I'm going to explain to you how it actually happens. Number 1, you need to locate a gene of interest. You have to know where it is, right? Which chromosome is insulin on, right? And within the chromosome, what's the locus? Where's the location? And then we are going to actually clone the DNA. Once you find it, you want to make many, many copies of it. You want to insert the DNA into a vector. A vector is something that will deliver the DNA to, say, a bacteria in this case. And then you will let nature do the rest. So this is a culture of bacteria. Some are blue, some are white. The blue ones are the ones that have accepted the vectors. Right? We can pick them up, and then we can culture them. Okay? We can mass produce them and turn them into little insulin factories, essentially. So let's start with the first step, locating the genes. Now that is a very difficult thing to do in the past, because like how do you even start looking, right? However, after the Human Genome Project was initiated and completed, it is much easier. You can just go online and look for it. Okay? So the Human Genome Project was an international collaborative research program with the goal to complete mapping and understanding of all genes in humans. Right? So they wanted to figure out all the ACTGs in the whole body. They wanted to figure out what they do and what kind of protein they code for, and so on and so forth. So it started on October 1st, 1990, and completed in April 2003. When I was in high school, like I did a presentation in my biology class for the Human Genome Project. They were just starting back then. And, it has been 20 years since it has been completed, right? 2003. So this gives us the ability for the first time to read basically the blueprints that allows nature to build a human being. So there is something called the genome browser, and you can just Google this thing. So if you open a browser and go like human genome browser, okay, and then you can click on it. It's free to anybody who has an internet connection, basically, and you can search up any genes that you like. Okay, so this is humans by default, and where is the oops, I was just doing this yesterday. Oh, there it is. Okay, so we can search like a gene. So let's say we want to find out the gene that makes insulin, or what, let's say do you guys remember any gene that we learned in the whole class? In the whole course, I mean. From like the cancer stuff that we talked about, maybe. Right, like there is the breast cancer gene, right? BRCA BRCA1. Okay. So like you can type that in and then just click on it. And then it will show you, oh, okay, this is the BRCA1 gene. It is on chromosome 17 at this exact location. Okay. So these are all the different versions of it. We have the exon and the introns. Like we can click into it. And then you can ask for the sequence, genomic sequence, Right? Click on it. I'm just showing you, right, like how someone would do this. And you click submit, and there it is. This is the G. Right? G C T G A G A C T, and then it's just like pages and pages and pages of this stuff. This whole thing. This whole thing. That's the BRCA1 gene. Right? So you will know what it looks like. And if you want to clone it, that means you got to copy all this and make millions and millions of copies of this thing. And you can also do it for like the insulin gene. You can do it for any gene. As long as you know the name of the gene, you can do it. Do you guys understand? Okay, so this is the freely available online for all scientists who are doing research, like telomerase, right? That's another one that you can search up, right? That's another one we learned, right? And then it shows you, oh, this is on chromosome 3, right? Right here. Okay, and then you can click on it and you can find the sequence. All right, so that's how you locate it. That's how you locate a gene. So what's the next step? After locating the gene, you can clone it. And to clone a gene, we use the technique called PCR, which stands for polymerase chain reaction. Polymerase chain reaction. If you have the segment of DNA and you know and you know the ends of it, then you can make many copies in very short period of time. Okay. So you might ask, like, how do you know what the ends of the DNA looks like? That's why we need the genome browser, right? Like when we look up the gene. Hold on, let me just see if I can pull up another one. Just like a random one here. This was telomerase. Like you look it up online and you're like, okay, the N is GGG, TTG, CGG. That's one N, and then the other N is like, you read it backwards, right? AGT, CC, AAAC. So if you know the ends of the gene that you're looking for, then you can clone it. Okay, then you can clone it. I'll tell you how it's done. So you can buy a machine. It's called a PCR machine. And then you put the ingredients in a little tube, and then you put it into the machine. You set it up, and then you let it run. It takes very short time. Okay. You put it in, you set it up. The whole thing can be done within 2 to 3 hours, and you would end up having like billion copies of the DNA that you were trying to clone. All right, and if you're doing it very carefully, and your machine is really sensitive, then you can copy with just one DNA strand. Okay, you don't even need a lot of samples to start with. Right? So like if someone recover like a drop of blood from a crime scene, right? A drop of blood actually contains a lot of genetic material. You can clone those genetic material and match it with victims and suspects and whatnot. So how PCR actually works is actually based on our understanding of DNA replication. So we learned this earlier in this unit, right? Lecture 6. DNA replication follows the semi-conservative model where you separate the strands and then you use each strand as a template to copy the DNA. So how do you pull apart the DNA strands? Well, if you remember, we need to use these enzymes, right? The gyrase and the helicase to do it. So in PCR, instead of using these enzymes, we use temperature. Yeah. What's holding the two strands of DNA together? Right? What's holding the A's and the T's and the C's and the G's? It's the hydrogen bonds, right? Remember, there are 2 bonds holding A's and T's and 3 bonds holding G's and C's, right? So when you heat up the sample, the heat will break those hydrogen bonds and it will cause the strands to separate. So you need to heat it up to about 95 degrees Celsius. The temperature might change depending on your GC content. Okay, so again, like you look it up here, and you're like, oh, okay, I see a lot of G's and C's here in the sequence, right? Like you can actually calculate the percentage, and it will tell you how much energy, what temperature you need to separate it. More CGs, you need to turn it higher. If it's like a lot of A's and T's, then maybe it doesn't have to be as high. But heating it up is going to help separate the DNA strand. Once the strands are separated, each strand can be used as a template to build the complementary strand. So in PCR, we have to create the DNA primer, just like actual DNA replication. DNA replication, you need to have the primase to put on the primer so the DNA polymerase can start building from it. But in PCR, you have to create your own primer. That's why you need to know the ends of the gene. You're going to create a primer that binds to this, right? That has to be complementary to this strand, which would be like CCC, AA, CGC. And then you have to create a primer that's complementary on the other side. You're going to add it to your reaction tube, and then you're going to be able to clone it. So to build the strand, to build the DNA strand, you need to have polymerase. And again, back in the DNA replication, we needed to use DNA polymerase, right? DNA polymerase 3. That was the one. But in PCR, because we are heating up the tubes to like 95 degrees, if you use the polymerase 3, it's going to break down at that temperature. Polymerase 3 is designed, if you would. It's like it's adapted to operate at body temperature, which is 37. If you heat it up to 95, it's going to break down. So what happens is we have to use a different kind of polymerase called the tag polymerase. Okay, tag polymerase is extracted from bacteria that lives in hot springs. And you probably have seen like these hot springs picture before. The color they are because of the mineral that builds up. They are super hot, hundreds of degrees, and there are bacteria that lives in those hot springs. So their polymerase, it's heat resistant. Okay, so you would get extract the tag polymerase from them, and it replaces the function of normal DNA polymerase 3. So that's basically what it looks like. We have the template here, 5' to 3', and 3' to 5' on the other one. We use the heat to separate it, right, 95 degrees. Right, the two strands pulls apart. These are the primers. The red thing are the primers, which you design based on the sequence that you found online. And then the tag polymerase will continue building the rest of the strand, right, like this. So after one cycle, this is called one cycle, you will have 2 copies. 1 copy, 2 copies, which are identical to the original. Okay, so like if you do this 5 times, if you repeat this process 5 times, how many copies are you gonna be able to make? So 1 copy becomes 2, 2 copies becomes 4 copies, becomes 8 copies, 16 copies, 32 copies. So that's 1 cycle, 2 cycles, 3 cycles, 4 cycles, 5 cycles. 2 to the exponent 5 is 32. Okay, so if you do this 20 times, 30 times, if you plug it into the calculator, you literally end up having like millions and billions of copies. Okay. So there are 3 steps here in PCR. First, we have denaturation, which is what I told you already. You heat it up to about 94 to 96, and the denaturation is going to separate the DNA strand. That is mimicking the function of the gyrase and the helicase. Right? So instead of using gyrase and helicase, we are using heat as a way to separate the DNA strand. Okay, so that's number 1, denaturation. Number 2 is annealing. Right? you need to cool the temperature down a little bit. Okay. Cooling the temperature allows the primers to bind. Okay, remember, these are the primers that we have created. If it's too hot, right, they're not going to be able to bind, right? So you lower the temperature a little bit, not so low that the DNA strand is going to come back together. It's still a little bit hot, but just low enough for the primers to attach. Just low enough for the primer to attach. The like the temperature, how do you control the temperature? Is that what you're asking?
Yes.
Yeah, at the machine, right? So this machine, the PCR machine, like you put your samples in, you close the lid, and then you program it. You say, okay, it actually is very simple. It has like, I mean, depending on the machine, but most machine has like 3 settings. You tell them what temperature you want for denaturation, what temperature you want for annealing, and what temperature you want for elongation. You just key it in, and then the machine will heat up, cool down, heat up again. All right, it's a very smart machine. So you allow the primers to bind, and then you need to heat it up again, right? Heating again, heating the temperature, increasing the temperature, I guess, sorry, increasing the temperature will allow tag polymerase to copy the DNA. Remember, the tag polymerase is taken from these hot spring bacteria. So when you cool it down too much, they are not going to work. Just like if you heat up hours, it's not going to work, right? So for the bacteria, tag is different. They are adapted to work at high temperature. If you cool it down too much, they don't know what's going on. They stop working. So you need to reheat it to 72. And again, these temperatures are not like set in stone, right? Like, you can change them. But the idea is you heat it back up, you activate the tag polymerase, now you make the copy. You don't want to heat it up too much. You don't want to heat it up all the way back up to like 94 or 96, because then the primers will fall off, right? Okay. So look, you separate, you denature. All right, so that was what, like 95 degrees. You cool it down to allow the primers to bind, and then you heat it up again so that you can copy. Okay, you want to keep it at 72 for some time to allow the polymerase to build the DNA, right? Of course, you have to supply the nucleotides as well. And then afterwards, you have 2 copies. And then the 2 copies will undergo another round of denaturation, annealing, elongation. Then you have 4 copies. And then you go through another round. Then you have like 1, 2, 3, 4, 5, 6, 7, 8 copies. And so on and so forth. So each time you do this cycle, you double the amount of DNA that you have. And before you know it, you have a lot. Okay? So it's important to remember you're not copying the entire human genome, okay? You're just copying a segment of DNA. And you choose which segment to copy because you design the primers, right? Okay, like if you want to copy this gene, you're gonna have to design primers that match the end. If you wanted to do the other gene that I was showing you, which was like pages and pages long, then you need primers for those. Okay. Are we together? So we located the gene using the internet. We cloned the DNA, cloned the gene using PCR. The next step is to insert it into the vector. Okay, and that is going to take some time to explain. So let's go through some questions here. You start with 10 copies of a DNA fragment. If you clone that fragment using PCR, how many copies will you have after 4 cycles? What do you guys think?
60.
Okay, that's one answer. Anyone else? Thank you, by the way. Anyone else? You do 4 cycles. So 16 is partly correct. If you do 4 cycles, it's 2 to the 4, which equals to 16, right? Okay. But we started with 10 copies. Right? So that's why I have to take 16 and times 10, which is 160. Right? If you started with 1 copy, 1 becomes 2, 2 becomes 4, 4 becomes 8, and then 16. Right? But then we have 10 copies. So 10 copies becomes like 20 and then 40 and so on and so forth. That's why it's 160. All right. So before we take the break, I want to talk a little bit about the PCR that is being used in COVID testing. Okay. So when it comes to testing whether you have COVID or not, I'm sure like either you have done it yourself or like someone you know in your life has done it by this point, right? Like everybody has probably know someone who got COVID, right? If not yourself. So there are 2 kinds of COVID tests. There is the antigen test, right? Antigen test, which is the one that you can do at home. Okay? You stick the thing up your brain or your nose, right? And then you collect the sample, and then you do a little drop on that little tray, right? And then you see whether you have 1 line or 2 line. That's an antigen test. An antigen test tests for the presence of the viral proteins. Okay? Do you have the proteins that are produced by the virus? That's what it's testing. So, I mean, it's decent, but it's not like as sensitive as PCR. So, antigen test tests for viral protein, whereas PCR tests for genetic material from the virus. Right? So by now, I hope you know the central dogma of biology, which is like DNA becomes RNA, and then it becomes protein, right? That's the flow of information. And this process is called transcription. You know that already. So in the kind of PCR that is done for COVID testing is not exactly the same as the PCR I just described to you. This one has extra steps. So it's called RT-qPCR. RT stands for reverse transcription, and Q stands for quantitative. So this PCR is measurable in real time, and RT is reverse transcription. So what does that mean? Well, the virus that causes COVID, the SARS-CoV-2 virus, that's what it's called, right? It's an RNA virus. RNA virus. The viruses can be DNA virus or RNA virus. DNA viruses are less scary in a sense because they don't mutate as much as RNA viruses. So like the chickenpox virus, right? The chickenpox virus is DNA virus and it doesn't mutate. So if you got chickenpox once, you're not gonna get it again because your immunity is there, right? It will be able to fight off the same thing. If you get a vaccine for chickenpox, you're not gonna get it, right? But for RNA virus, they mutate very rapidly because they don't have a spell checking mechanism. So every time they copy their DNA, they make some mistakes and the mistake stays. And some of these mutation might be beneficial for them, right? Yeah. So that's why, the flu shot that you got last year might not work this year because it's a different strain of flu. And for the COVID virus, they keep on mutating, keep on changing. So even you got COVID once, you can get it again. Same thing with the vaccines. So when you extract the sample, because it's viral RNA, you have to change it back into DNA before you can do the PCR. So the opposite of transcription is called reverse transcription. You change the RNA back into DNA. That's what this RT is. Okay? So once you've done that, you can actually get a different machine. Not only does it do PCR, but it's able to track the number of copies in your sample in real time. And then you can plot it on a graph. Right? On the x-axis is the number of cycles, and on the y-axis, I know these numbers look weird. That's because it's like a logarithmic scale, but doesn't matter. Okay, just think of them as like number of copies. The higher up it is, the more copies you have. That's all. So there is a threshold that people set, right? Let's say 200 copies or 2,000 copies, whatever the case. You are trying to figure out how long does it take for your sample to create that many copies. Okay, so each of these lines represent a sample. And so, let's say this one, let's call that A, and then let's call this like B. All right. So sample A reaches the threshold, in around 12 cycles. Sample B takes about 22 cycles to reach the threshold. Again, the threshold is just a number of copies that you interested in, 2,000 copies, for example. Okay. So I want to ask you, which person has a higher virus load? Okay, which person has a higher viral load? Person A or B? Who has more viruses in their body? That's my question. Is it A or is it B?
Looks like A.
That's right. Because you get to 2,000 much quicker than B, right? All right. If you started with a lot of samples at the beginning, then you will be able to copy it much quicker. Right? So this is useful for contact tracing, like early on in the pandemic when they were still doing it, right? Because the person was getting it, was having a higher viral load, he or she probably recently caught COVID, right? This one, not so much viruses in them anymore, right? He's recovering, right? It's getting better. All right, so that's RT-qPCR. When we come back, we're going to talk about how to put the gene into the plasmid and feed it to our bacteria. All right, let's take 5 minutes. We'll resume at 9:45. If you have questions that you want me to take up from lecture 8, those genetic problem, just type the question number in the chat box and we'll do it afterwards as well. All right, see you in a bit. Okay, welcome back. Let's keep going. Let's recap what we're trying to do here. We are trying to learn about recombinant DNA, which is trying to put DNA, genetic material from different organisms into the same thing. And we are trying to understand how to use recombinant DNA technology to to create insulin, basically, right? So that we can give it to people with type 1 diabetes, right? So we located the gene using the genome browser, which was created from the Human Genome Project. We are able to clone the DNA that we want using PCR. So in this case, we will clone the gene that will allow you to make the insulin. Okay, so now we have millions of copies of these genes that has the instruction to make the insulin. And now we're gonna have to put it into a bacteria. So just to refresh your memory on the bacteria genetic material, they have their own circular DNA, which is shown in red here, but they also have additional little circles called plasmid. Right? These plasmid can be exchanged between bacteria. Sometimes they like to pick it up from the environment, and usually the plasmid would give the bacteria some kind of survival advantage. It maybe allows them to make an extra shield, capsule, we call them, to protect them from drying up. Maybe allows them to create little hairs that allows them to stick together to form a biofilm. There are different features that can be acquired from these plasmid. Okay, so the idea is to give them a plasmid that contains the insulin gene, excuse me, that contains the insulin gene, and then the bacteria will be able to use that to make insulin. But first, we have to put the gene in the plasmid. And to understand how to put the gene in the plasmid, we have to understand this concept of restriction enzymes. Bacteria produces these restriction enzymes. They are kind of a defense mechanism against foreign genetic material. And they don't like to have foreign genetic material going into them, like from a virus, for example. And these enzymes will cut the DNA that are coming in. It's not going to cut their own DNA. It's not going to cut the plasmid because of reasons we're not going to go into. But the point is, it's selective. It knows what doesn't belong there, and it's going to shred them up, basically. And the way it does it is to recognize sequences that are 4 to 8 base pair long, and they are usually special because they have complementary palindromic sequence. So what does that mean? Well, you know what complementary means already, right? Like ACTG, the pairing rule. But palindrome, in English, a palindrome is a word that reads the same forward and backward. Right? So like madam, that's a palindrome because if you spell it forward, it's the same as when you spell it backwards. Right? Level, wow, kayak, all these are examples of palindromes. This is a really long palindrome. Was it a car or a cat I saw? If you read it backwards, it's the same. Was it a car or a cat I saw? Right? So palindrome, same forward and backwards. So in DNA, a palindrome is something that looks like this. GAATTC. And on the other strand, it's complementary, of course, but it's the same when you read it backwards. GAATTC. Okay. Forward and backwards the same. And then this would be a restriction enzyme. It will find these sequences and then it will cut it. So you end up having 2 pieces. Do you understand? So I can create another palindrome to show you, right? Like ATT, AAT. So the complementary strand would be TAA, TTA. But if you read it forward on one strand, it's the same as when you read it backwards. So these are called palindromic sequences. Okay. These are the different bacteria that we can extract the restriction enzymes from. These are not the only ones, okay? There are like many other examples, but these are the common ones. So the enzyme is called like EcoR1, which is extracted from E. coli. Right? We have a bunch of other ones from different bacteria. So here is the recognition site for each of the enzymes. So for EcoR1, they recognize the GAATTC, and then they would cut it. Right? The arrow tells you where the cutting begins. So it cuts through the G sideways like this. Okay? And you have that as your product. Sometimes it just cuts straight down like this. Okay? So these ones, these 2 here, because they are cut straight, right? They create what's called blunt ends. Blunt ends. On the other hand, these ones, where it creates like a zigzag cut, these are called sticky ends. Sticky ends, because they can come back together, right? Like the AATT over here, that can actually bond with a TTAA, and then they could just like reattach. It's like Velcro. You rip them apart, they could come back together. So for the purpose of inserting a gene into the plasmid, we want restriction enzymes that can create sticky ends. Okay, let me repeat that one more time. we are trying to put a gene into a plasmid, right? So we need restriction enzymes that can create sticky ends. But first, let's do a sample problem. There is a restriction enzyme here called SMA1, and I give you the recognition site, GGGCCC, right? And here is the DNA sequence. So if I treat it with SMA1, why don't we figure out how many times it will cut it? So look, this is from 3' to 5'. So I must be careful. 3' to 5', I'm looking for CCC GGG. So I just scan the sequence for CCC GGG. There, CCC GGG. That's the same sequence that it recognizes, right? Okay, which means it's gonna cut right here. Right, the arrow shows you where to cut, right? So that's one cut. Now we're gonna keep scanning and see if we can find another CCCGGG. There it is, CCCGGG. All right, so it's gonna cut it. So 2 cuts, 3 fragments. Okay. Identify the palindromic sequences in the following DNA fragment. This is just one strand. You don't have to write out the second strand, but you want to try to find the palindromes here. So let's take a look. For example, over here, I found one already. GCGC. The complementary strand backwards would be GCGC. So there is a palindrome. Okay. Remember, these are 4 to 8 nucleotides long. So please go ahead and look at the strand, see if you can find another one. GGAA, well, GGAA is not quite it, right? Because if you put GGAA, the other strand is CCTT, right? So if you read it forward, it's not the same as if you read it backwards, right? But good try. Does anyone else have another palindrome? All right, here's another one. GCAT TCG. So let's maybe not the G. Okay, hold on, right here. CAT. Actually, you know what? That's not even it. It's over here. Right here. Okay, let's write it out. G pairs with C, TTAA, G. So if I read it forward, it's GAATTC. If you read it on the other strand backwards, GAATTC. Right? That's another palindrome. Okay. Another one right here, AATT. And then another one here. Okay. That's it. So those are the palindromes. And if each one would probably have a different restriction enzymes for cutting it. Here's another question. Please give it a try. We have two restriction enzymes here, equal R1, which recognize this particular sequence GAATTC, and then we have HIND3, which recognizes this particular sequence, and we have a DNA strand here, and we're going to be treating it with both restriction enzymes. So how many fragments are going to be produced? I'm just going to give you a moment to try this out. I'll be right back. All right, anyone have an answer for me? How many fragments are we going to see? 4? All right, let's see if you're correct. So we have GAATTC. So here, GAATTC. That means you're going to have a cut here, right? It's going to cut from the G to the G like that. That's the equal R1. And then GAAT, is there another one? Right here, GAATTC. So that's another cut. Excuse me, right there. And that's it. Let's take a look at the second one, HIND3. And that is AAGCCT. Let's see, AAGCCT right here, AAGCTT, right? So it's going to cut like this, and that's the only one. Okay. So 3 cuts, 4 fragments. 1, 2, 3, 4. That's the correct answer. Very good. All right. So now we want to know how to put this into the plasmid. Okay? And to do that, we're gonna have to create the sticky ends. Right? So you can see if you cut it with the same restriction enzymes, then the fragments can stick together. Right? Over here, the red one here shows that it's a HIND3 fragment, which is ATTCGA, and that's not compatible with the equal R1 fragment. Guess we can draw it out to see. Okay, and draw it out to see. So here we go, inserting a gene into plasmid. All right. Now to keep it simple, I am just gonna say there is a restriction enzyme called restriction enzyme X. Okay, restriction enzyme X. And the recognition site is AAATTT, just to keep it really simple for us. Okay. So like, afterwards, after the cut by the enzyme, you're gonna get this. Do you understand? Now I want you to take a look for a second, if you are not already looking. Like I can actually put it back here, right? And then they will match, right? I'm gonna make it back bigger, I guess. You see? I can put it back together, right? And they're complementary and they will stick back together, right? So that's what sticky and means. All right. Now I'm gonna create another one, let's say restriction enzyme Y. And this one is CCCGGG. Again, just to keep it simple. And then after the cut, you're gonna have C. Right, I'll show you where the cuts are as well. Right, you will have C, G. There's an extra one there. There's actually an extra one here as well, sorry. Right? Yeah, something was wrong, but now it's fixed. Okay, thank you. So here's the point. Only the sticky ends from the same enzyme can stick together. I cannot take this red piece. I cannot take this red piece and stick it to the blue, right? You see, it's not compatible. The C and the T's don't match. It won't work. So that you can't stick them together. You can only stick them together if it's cut with the same restriction enzymes. All right. Okay. Now we are going to buy a plasmid. Okay. Plasmid you can buy online. So here's a plasmid. And just again to keep it simple, in this plasmid, when you buy it, it will tell you the exact sequence of the plasmid. Okay. In this plasmid, there is going to be AAA, TTT here. And from PCR, we have created a gene. We have, cloned a gene. We make many copies of the gene. Right? This is like insulin or something, insulin gene. Right? And it happens that at the end, you also have the same sequence. And since you can search up the sequence online, you'll be able to figure it out exactly, right? You just have to make sure that the sequence that we're trying to cut does not show up in the middle of the gene. Otherwise, you're going to cut the gene, right? So this is what we're going to do. We're going to treat this plasmid with restriction enzyme X. Right? So it's going to cut right here and right here. I'm going to also treat this with restriction enzyme X. So it's going to cut right here and here, right here and here. All right. I mean, you don't really have to draw this now, right? Now you can just watch and understand, and then you can copy it when you watch the recordings, right? So here is gonna do this cut. So what do we have? Well, we're gonna have our plasmid, but now it has been cut open. It's been cut open, and I'm gonna have this on one end and this on the other end. So I have created the sticky ends, right? Sticky ends. If I do the same treatment for our gene, you're going to create the same cut. Create the same cut. And we will have the gene. And we will have this. Like the other half just kind of falls off, right? I mean, we don't really need that. And same thing over here. Okay. And then the other half, the bottom half, just kind of falls off. We don't need it. And so we have this part here. And we have this part here. Okay. The other half just kind of falls off. We don't need it. Okay. So if we put these together, and then I add the glue, before you remember the glue is ligase from DNA replication, then this gene is basically going to, let me see if I can do this here. This gene is basically going to go into the plasmid like that. Okay, that's not very good. But you can think about it. Look, these are sticky ends. And then these are also sticky ends, right? Because they are being cut with the same enzyme. So afterwards, what you're going to end up having is this. You are going to have the plasmid with the gene on the inside. That's how it's done. Okay. And then you're going to feed this to bacteria. So that's your bacteria, and now it has the plasmid, right? It will turn into an insulin factory, basically, right? Any question? So, one last problem. How do you know if the bacteria have taken up the plasmid? Right? Like, I mean, they would look exactly the same under the microscope, right? Just like if you look at a person, you can't see their DNA, right? So, like, you know, how do you know if the bacteria has accepted the plasmid or not? So, that's where we have to learn the next thing. So when you buy the plasmid, this is a very simplified plasmid, okay? So when you buy a plasmid, it will tell you what's on the plasmid. So typically it has these things. We have the restriction enzyme sites. Right? So it tells you where you can insert the gene. You can insert the gene over here if you use ECOR-R1. You can insert the gene here if you use PST1. You can insert it here if you use BAMH1, right? Like, multiple location. And why would you use one restriction enzyme over the other? Well, there are many reasons. One reason is this. When we use the restriction enzyme to cut our gene, we have to make sure that the sequence does not show up in the middle of the gene, right? Let's just say for the sake of discussion, like if inside our gene there is another AAATTT, then the restriction enzyme X is not suitable because not only are you cutting the ends of your gene, you're also cutting within your gene, right? So you might have to use restriction enzyme Y, right? So if the plasmid has multiple insertion sites, then it just makes it more versatile, right? So then we also have these other sites, these other markers. Okay, AMPR stands for ampicillin. Ampicillin resistant gene. Ampicillin, of course, is a type of antibiotics. And over here we have tetracycline resistant gene. Okay. So let's keep it very simple right now, and I'll explain more next class. All right. Let's say you inserted using ECOR-R1. You inserted a gene using ECOR-R1. Right, so this is the bacteria. You're gonna feed the plasma to the bacteria. But again, I don't know if they accept it. Okay. Some of it will accept it, but some might reject the plasmid. They don't take it up. All right. So what will happen? Well, let's see. This is the plasmid, and it has the gene in it. Okay, it has the gene that we want in it. But along with the plasmid, it also gives them ampicillin resistance as well as tetracycline resistance, right? So if I grow these bacteria, grow in the presence of ampicillin. So I give them the nutrients they need to grow, but I also add a little bit of ampicillin in the culture. What will happen to the bacteria that does not have the plasmid? Well, if they don't have the plasmid, if they did not accept the plasmid, they will not have ampicillin resistance. They will die. But if they accepted the plasmid along with the gene that we want them to take, they would have the ampicillin resistance and they would grow. Right? So at the end of it, you look at the plate, the bacteria that survived, that are able to grow despite having ampicillin in the culture, those are the ones that we want. Those are the ones that have accepted the plasmids. Those are the ones that are going to be producing the insulin. Okay? So I'll expand on that again next class. Like I said, right? Like the quiz is difficult because of all the problem solving. So make sure you work on those problems, okay? And start studying for lecture 9 as well. We are halfway. I think we will definitely be able to finish next class. And we'll keep the quiz for Tuesday next week. That's all. Thank you. Enjoy the rest of your day. Bye for now.